Hi, I'm Brad Power, and this is the Cancer Patient Lab, and this is our weekly webinar series. Today, we're pleased to have with us Kaumudi Bhawe, who's a longtime collaborator in this space. She'll expect to introduce herself, but she's a PhD microbiologist. We got to know her originally through her work at Cancer Commons. She's now doing other independent things, which I expect she will also describe. But she offered to help us with some of the basics of introductions to the hallmarks of cancer. And the hallmarks of cancer, for those of you who don't know, are the basic attributes of cancer cells and how they propagate and so on. And we really haven't had a session that's done this kind of basic introduction. And because it's quite a number of topics, how many hallmarks are there now, Kaumudi? Like a dozen, 14?
Also, there's 10 in the circle, and then there's several factors. I'll get to that in the talk. We'll
Yeah.
kind of progress through it. So
The point being, you could easily spend many sessions talking about them. This is the first. If need be, we'll have another. But she used a survey to decide which would be the most useful ones to talk about for our audience. Let me just cover the typical few housekeeping things we have at our outset here. This is for information purposes only. The purpose of the Cancer Patient Lab is to arm patients and caregivers with information they can take to their medical team so that they can be full co-pilots with their medical team in their care. So it is not medical advice. The second is that we are making everything public. So if you're concerned about your image being made public or anything, turn off your camera, change your name, and don't say anything. And then finally, we are a volunteer nonprofit organization, so we depend on the kindness of donors and would welcome any donations you might make to our nonprofit organization, which you can do through our website. And with that, I'll turn it over to Kaumudi. I already did the introduction, but basically she will take us away and help introduce us to some of the basics of the disease of cancer.
thank you so much, Brad. Thank you, everyone, for being here. And thank you, Brad and Cancer Patient Lab, Brian, all of you for giving me the opportunities to speak here today. So as Brad mentioned, I have a PhD. I have a PhD in public health with a biostatistics data science driven project in brain cancer, actually. But I also have a background in biotech, 15 to 20 years of experience in various roles, ranging from very early research up to late phase clinical development, essentially as in the clinical science team. And today I'm here to talk about the hallmarks of cancer. I recently, early this year, I have started a sort of I call it a passion project venture sort of thing. It's called Yom Wellness. And I will be introducing Yom Wellness to all of you as part of my talk. And I just wanted to say that our talk will have one or two sort of mindfulness coming together kind of activities incorporated within it. So those are optional. If any of you don't want to participate, that's fine. They're going to be short, very short, but just to give you a flavor. And with that, I'm going to, also, if there are any acronyms, any sort of terms that you have questions about, now it's so easy, right? You can just enter it into ChatGPT, Google, wherever. You can put it in the chat box in our talk. That's totally fine throughout the talk. If any of my slides are, you have those quick questions about the jargon or the acronyms, I'll try my best to sort of talk about those and talk you through those, but still. All right, with that, I shall share my screen. Give me a moment to share my screen here, and then I'm going to put it on a slideshow sort of format. Let's see. Do this slideshow. All right. And here's the use the laser pointer here. It's okay. You can see my, I hope everyone can see my cursor. So, all right. So our talk today is what patients and caregivers should know about the hallmarks of cancer. just an overview. As I mentioned, we will have a couple of short mindfulness activities interspersed. But an overview of what I will be covering today. One quick slide about Yom Wellness, which I talked about. Then the survey results. Thank you to all of you who participated wholeheartedly in the survey, which was so amazingly designed by our very own Dr. Frank Nothaft, and with input from Brad and myself. So we had several questions about what you would like to see today, and I tried to tailor my talk towards that. So we will go through the survey results really quickly, some of them. Then I will cover some basic molecular biology concepts that we will use throughout the talk. I'll share some of the very latest scientific research on tumor evolution, which came out actually October of this year. And then we'll go through the hallmarks. As you all know, there have been 3 landmark publications or papers on the hallmarks of cancer. All right. We'll talk about immuno-oncology, oncometabolites, treatment possibilities once cancer is diagnosed. And then now, how the field is moving towards AI-driven treatment recommendations in precision oncology. We'll have some points to ponder at the end. So what is YOM? YOM stands for your own meaning. And this is a science-based healing system, but it is a healing system for aligning the mind, the body, and the energy that we carry in this mind-body vessel of ours. This technique helps you to understand and express your own meaning so that you're able to regain the loss of equilibrium. So any diseased state where dis-ease, loss of ease, that happens because of a loss of equilibrium. And I'll get into that, the scientific basis for that very shortly. But we work with you to jointly explore what your own meaning is from all aspects, from mind, body, spiritual, all aspects, to sort of see how that affects the flow of energy within your body and how that affects metabolism, how it affects movement and capacity to affect change. These are breath and movement-based interventions designed to empower you, work in hand with the science that is known up to date. Just my website there. So now the survey results. We designed this survey for everyone in Cancer Patient Lab, and what did people have to say? So we got a total of 18 responses, and most people would like to understand the hallmarks to help in making informed decisions about next steps for treatment approaches, for testing approaches. Okay. And then we have a good mix here. We have a large, as we all kind of are aware, we have a large membership of people facing prostate cancer, some people facing pancreatic cancer, some who are facing brain cancer, and then the other cancers are less represented in Cancer Patient Lab. And then just quickly going through these. So we asked about the initial 6 hallmarks that were published in 2000 and asked people whether there was a specific hallmark they were more interested in. Across the board, most people are interested in learning about all of the landmarks, initial ones, except maybe a few people are more familiar with cell proliferation, for example, or how cells escape or are able to replicate and have immortality. Maybe people are more familiar with those hallmarks, but overall, most people wanted to sort of were interested in all 6 initial hallmarks, as well as we asked about what other hallmarks people were interested in. And the top 2 that came up were avoiding immune destruction and tumor promoting inflammation. So I will be talking about these topics as well. Okay, so before we get into the actual scientific meat of the talk, let's look at some very basic molecular biology concepts, which, and if you're already very familiar with these, then just bear with me. But this is just to bring everyone on the same page. Some very basic molecular biology concepts. Here, first, I just want everyone to spend a moment to look at the structure of the mammalian cell, okay? And you have the nucleus, you have the cytoplasm, and you have various cell organelles that are doing different functions. And all of these have molecules within them. Okay. Now, within cells, you have DNA, RNA, and protein, and many other smaller molecules as well. But the central dogma of molecular biology has always been that the information flows from the DNA to the RNA to the protein. And remember that this DNA is located in the nucleus. The RNA can travel from the nucleus to the cytoplasm, which you see here in this diagram, right? And then there's protein, which is present in the cytoplasm. One thing that's important to know about is that these organelles called mitochondria are the energy powerhouses of the cells. And that's where basically in a non-cancerous typical cell, energy processing happens through what is called the TCA cycle or Krebs cycle. But I'm not going to get into those details. But I just wanted to mention that here. Another thing, important thing about the mitochondria is that the mitochondria have their own pieces of DNA, mitochondrial DNA, and it's all maternally inherited. Another thing to keep in mind as we go through these slides and this talk is that there's a hierarchy in biological organization. So you can see levels of organization happening from atoms that get organized into molecules that are organized into cells, or larger molecules actually first, but then cells that get organized into tissues that further get organized into organs and systems, organisms, populations, communities, and so forth. So keep that in mind in the back of your mind when you're thinking of all the complex concepts we deal with today. Okay. And again, I'm giving you this talk with a lot of information, and you can consider it as a first talk, and we can have more if people want more. But I'm going to cover a lot of information. So throughout the talk, keep in mind that the biological organization, atoms, molecules, cells, tissues, that's happening, and it's actually happening in real time, dynamically as we speak. We don't think about this enough, but those atoms, molecules, cells, tissues, this organization is dynamic. Okay. Again, another very important concept is the concept of homeostasis. So what is homeostasis? Homeostasis is any self-regulating process, right? That it's a way in which biological systems are striving to maintain equilibrium. And again, that equilibrium is a dynamic equilibrium. And what do we mean by dynamic? This you see this kind of picture of a seesaw, kind of. It's tilting one way or the other, but it's trying to maintain stability on that fulcrum here. And you know, each time you have a stimulus that produces some changes in some variables in the system is trying to balance itself. And until the imbalance becomes too much and it's not sustainable anymore. So the conditions, when we say dynamic equilibrium, the conditions largely, if you break this down in a differential manner, like really small infinitesimal slices of time, the conditions are remaining largely uniform. They're going to be tiny changes, but change is the constant that's happening. So it's constantly trying to maintain that equilibrium. Keep that in mind as well. Okay. One pause, one breather, three concepts: structure of cells, nucleus, cytoplasm, organelles, mitochondria, energy, pieces of DNA, the nuclear DNA translating to RNA, translating to protein. All of this is happening in real time. Okay. In all the cells of our body. Those cells can be mammalian cells. Some of those cells can be the gut microbiome, in which case there would be no nucleus in those cells, but there's some sort of a dynamic equilibrium happening there in crosstalk. There are levels of organization happening. So let's take just about less than a minute, actually, to just kind of, if we want, just close our eyes, and each of us can go within and seek our own meaning, your own meaning that is so much deeper than the cancerous cells that it may be dealing with at any given point in time. And I say this scientifically because your own meaning, that entity which you call as yourself, the one that I'm calling Kaumudi, for example, began when it got a name as a baby or even maybe before that, way before that, began as a single cell maybe, and continues, right? The physical aspect of it continues. There is a slice of time or a chunk of time in which that collection of cells is dealing with a smaller, dysregulated collection of cells, which we term cancer. Keep that in mind. And now we will move to some of the latest scientific research on tumor evolution, how these tumors are evolving, trying to maintain their dynamic equilibrium in time, right? Before we do that, an important concept. One thing why I call this a workshop is because I would hope to also transmit or help people who are struggling to read very dense scientific literature. Now, of course, you have help of LLMs that are going to help you ease it down, but still, whenever you look at a paper or any sort of news, for example, any news about the latest scientific breakthrough, just remember this evidence hierarchy, okay? At the very top, going from bottom to the top is the gold, I mean, the best of the best, meaning rather the the most scientific evidence that is contained. And that's when you have a systematic review of randomized controlled trials. So hang on. What is at the bottom of this pyramid? You have case studies, you have anecdotes, you have information that's passed from one person to another. You have lab bench studies that are done in cells, that are done in animal models, etc. And you have personal opinions from professionals. It could be educated opinions, but that's what you have. It's one off all of these little small bits and pieces of data coming through. You on above that one step is observational studies that are done in a controlled manner, but they're cohort, they're case control, they're still smaller number of people. Okay. Then you have other controlled clinical trials, phase 1, phase 2, which are designed specifically to answer certain questions, but they're actually designed to look at safety, not efficacy necessarily, but they are nowadays more and more designed to look at early signals of efficacy that may be showing through even in smaller numbers of patients. You have the gold standard, which is randomized controlled trials. And on top of that, if multiple randomized controlled trials have been done for a particular intervention, then you have the systematic review. Okay, now, remember I said we're going to look at some latest scientific research on tumor evolution. What I'll show you is very early research material. But on 30th of October in the Nature portfolio, there's a series of 10 papers that talk about something called the Human Tumor Atlas Network. I just want all of you to be aware of these things that are happening on the forefront of research, because now more and more bench and bedside is becoming kind of more and more collaborating in real time. So the Human Tumor Atlas Network explores tumor evolution in time and space because these technologies are available to look at single cell RNA sequencing or proteomics, etc., in space and time. So you capture different time snaps of the tumor and you look at different locations, because as we all know, there is heterogeneity, there is non-similarity in different pieces of tumor and over time as well. So there were 10 papers in this portfolio that came out, right? One thing to note here is that these are mostly breast cancer and colorectal cancer studies for multiple reasons. Perhaps we won't go into those, but these 10 studies mostly focused on breast cancer and colorectal cancer. But I'm going to take one paper out of these 10 and kind of break it down a little bit on how one could look at a scientific paper like this, which is very research driven, okay? So you take this paper, Progressive Plasticity During Colorectal Cancer Metastasis. Now, here in the introduction, what are they saying? They took a cohort of biospecimen trios. So from a single patient, from the same patient, they're taking samples from the normal colon, unaffected colon, the primary tumor, and then metastatic colorectal cancer. Okay, so the metastases. And what they have shown in this very early research sort of paper is that although the primary tumors are largely adopting a certain stem-like state, the metastases adopt progressive plasticity, meaning there is large differences in the way they kind of diverge from that stem-like state. Okay, so they initially reprogram, meaning they lose their differentiation into as colonic intestinal cells, and they become more fetal-like or more embryonic, but then they start differentiating and adapting in multiple different ways. And how did they find this out? They took samples from 31 people, 31 patients, and with microsatellite stable. So these are all going to be now very technical terms, right? Microsatellite stable, mismatch repair proficient. So it's not likely to mutate as quickly as something that would have been called having microsatellite instability or is MMR deficient. So anyways, you have this relatively stable colorectal cancer, and they did this kind of surgery with, they removed pieces from normal primary tumor and metastases. Okay, and then these patients had different, some received no treatment, some had standard chemo before surgery, and they looked at this kind of collection of tissue. And then they collected additional tissue at a different time point. Metachronous just means at a different time point for 6 of those patients, right? And now just look at the picture here. We're not going to go into the depth of this, but what did they do? They just show here, you see the process, right? 3 different locations, and then they're doing something called single cell transcriptomics. So they're looking at all the RNA transcripts. Remember I showed you DNA to RNA to protein. They're looking at all the RNA transcripts, sorry, that are measurable to date. And they're using bioinformatics, data science type of tools to cluster those into groups of transcriptomic expression, or this RNA expression that is known in the literature to represent these different type of cell types or phenotypes, if you will. Okay. And when they say canonical, that means something that's known for a longer time. Non-canonical is something more surprising, more recent findings. You can see that there's a wide distribution across the different types of patients, and they have just kind of looked at that and studied how it correlates to survival. They've looked at this from in a retrospective manner from TCGA data, as well as another database that they had. Okay, so that's extremely complex, full of jargon, and very early research. One single paper. There's multitudes of research studies that are ongoing right now. Okay, but how does this help you, and how is this related to the hallmarks? So we go to hallmarks. We begin with hallmarks in the year 2000. Okay, in the year 2000, that's about 24 years ago now. Dr. Hanahan and Dr. Weinberg came together and said, there's all these studies coming out of so many different labs. Let's put this material in a manner that can be sort of understood. If we're trying to get to, I'm going to put in air quotes, the meaning of these tumors, what are they trying to do here? Let's put them into 6 different principles. What are tumors across the board, across different types of cancers, across different, phenotypes and whatever is seen in the clinic? What are these tumor cells trying to do? They're trying to keep growing, keep proliferating, keep evading cell death. Okay, they become insensitive to anti-growth signals. They pull in blood vessels to keep themselves sustained. So sustained angiogenesis, they actively secrete factors that are pulling in the blood vessels towards the tumor. They somehow gain this limitless replicative potential, and then they're able to invade other tissues, right? And at that time, just to put things into perspective, the Human Genome Project was completed in the year 2003, where 92% of the human genome was sequenced. And just the pace of things, the final complete sequence was published in the year 2022 in the issue of Science. It didn't receive as much fanfare and news coverage as the initial Human Genome Project, which covered only 92%. That's large amounts, but still, the remainder was done. Just have that perspective. And so at that time, they said, okay, here's an emergent integrated circuit of the cell. So how are these cells using the molecules within them to, how are they able to bring about these changes in themselves and the surrounding cells? So at that time, you saw this rudimentary diagram, right? With in the nucleus, there are changes in gene expression that enable the hallmark capabilities. You see some familiar molecules to all of you over here. You see P53, you see MYC, you see KRAS, RAS here. Okay, RAS is in the cytoplasm. So the signaling is happening both ways, from outside growth factors and proliferation signals that are coming from the extracellular matrix into the cell through the cell membrane and into the nucleus, and then from the nucleus back out to the cell membrane and outwards into the extracellular matrix, which then sort of does this crosstalk across tissues, right? So a few things that these signals are saying to these tumorous, cancerous cells, they say, keep dividing, don't die, stay flexible, and keep moving. Okay, but using all these molecules to do that. And then at the time, now it seems so primitive or rudimentary, but it was revolutionary at the time. But then they said, okay, here's a reductionist view, which is looking at this lump having just cancer cells, but we really need to think about what else is there as well. So there are immune cells, there are fibroblasts. We need to be thinking about that as well, a heterotypic cell biology. But that's the year 2000. Flash forward 11 years later, they had to revise the hallmarks. They had to come up with more that would explain what's happening in these tumors as more evidence was collected. And so the next generation, what they did was they had the initial 6 hallmarks, but then they added 2 emerging hallmarks that you see here. Is everyone able to see my cursor? Yes, just like raise your hand. Okay, okay. So they added 2 emerging hallmarks and then 2 enabling characteristics. So in addition to evading apoptosis, maintaining this proliferative state, and all of, pulling in angiogenesis or blood vessels, all of these 6 things that they had identified that tumors do across the board, they now said, oh, wait, we are dating this and saying there's 2 more. There's 2 more. There's genome instability and mutation. Okay, and this now we can say, because so many of you are aware of this microsatellite stable versus unstable. And also there's cancers that are more within this category that have more instability and more mutations, and then there are some that are less so. Okay, it's all dependent on tissue characteristics, on the tumor characteristics. So another enabling characteristic is the ability of that tumor to promote an inflammatory sort of a environment around it. Okay, that's another enabling characteristic, keeping that tumor in place and allowing it to sort of become stronger. And then some emerging hallmarks that they said. So enabling characteristics of those tumors and emerging hallmarks, they said, yes, these tumor cells are deregulating cellular energetics. So remember, I pointed to the mitochondria in a normal cell, in a healthy cell. There's glycolysis, which is the processing of sugar. But then there's a longer procedure called the TCA cycle or Krebs cycle, which produces more ATP or more energy in the cell. But then cancer cells somehow kind of twist that, and they don't rely as much on the mitochondrial oxidative phosphorylation based process as much. They rely on a kind of a deviated form of glycolysis. And there are people actually studying diet, lifestyle changes, and all of that in conjunction with also medications to intervene where the cellular energetics of those tumors has kind of gone away. Maybe there's ways that can be affected positively. And then those tumors are trying to avoid immune destruction. So now in the age of immuno-oncology and past that age almost, even where it's become a stable part of a lot of regimens, we know that. But in 2011, remember that this was before immuno-oncology really took off at all. And so they said that, okay, an emerging hallmark is this avoiding immune destruction. And now here you will see, okay, there in this left-hand corner, you will see that their diagram of how a tumor looks became more elaborate than was initially what they had depicted. And they did a really amazing work here to kind of put this in a diagrammatic format where you see if there's if the tumor is sustaining proliferative signaling, then there's at least a group of tumors that respond to EGFR inhibitors. For example, they're just giving an example of the kinds of treatments that sort of are geared towards or targeting these specific hallmarks that they have identified, right? You have PARP inhibitors for if there's genome instability and a large number of mutations. You have, of course, inhibitors of VEGF signaling, which were some of the earliest remarkable drug approvals at the time, but inhibitors of VEGF signaling if the tumor is inducing angiogenesis and you want to sort of attack that. So there's this whole array of treatments, but now we're in the age where we have to look at how can they be combined, and if there's more needs, lots more needs to be done. So in this 2011 note, they in fact said that clinical responses have generally been transitory, right? Because they're followed by the tumor adapts, and there's some cells maybe that just hang around and were not detected, and the tumor changes because of that dynamic equilibrium that it's trying to maintain. And so they also made this insight here that was coming in the field that each of these core hallmark capabilities is regulated by partially redundant signaling pathways. We know this now, yes. Given that the number of, again, I've highlighted here, given that the number of parallel signaling pathways supporting a given hallmark must be limited, it may become possible to target all of these supporting pathways therapeutically, thereby preventing the development of adaptive resistance. That's a great thought. We have to figure out how they'll be combined without combining the toxicities synergistically in a bad direction. So that has to be figured out by science. And now fast forward to Hallmarks 2024 of this year. What has happened with Hallmarks 2024? We have to embrace cancer complexity. We have to embrace cancer complexity, meaning now that wheel, that circle that you saw with the genome instability, the increased vasculature in the tumor, activating the invasion and metastasis pathways, all of this is there, yes. But we have to remember that all of this in those tumors, okay, think about this for a moment. At any given point in time, even in the scenario of the most aggressive and highly treated cancer, the number of, this is an encouraging thought, but this is still I'm not trying to give some sort of new age, happy, everything's hunky-dory sort of a feeling, but a realistic view, a scientifically based realistic view, that even in the most aggressive, highly treated, highly spread cancers, it is true that the number of cells that are not actively cancerous or tumor cells is more in number than the cells that are in the tumor and around the tumor and affected by the tumor. Okay, just remember that always, because that is what the scientific field is saying in a scientifically jargon-laden manner in the top journals. These are at the top of that evidence pyramid that I showed you. This is synthesizing the information that's coming out of those lab bench studies, out of clinical studies, out of everything. At the top of that pyramid, you see the most amazing synthesizers in the field. I mean, they're more than that, but okay. They have put this together. They're saying that, yes, you have this wheel of those hallmarks, which happens to be, I think one of the previous speakers had used her word as terrain. So I'm going to leave that as her word. But it happens to be in this milieu or this larger environment of, you have polymorphic germline susceptibilities that are in maybe in an individual. You have mutations that could be caused by environmental agents. You have the larger environment. You have metabolic effects that can be affected by diet, exercise, many different factors. You have aging systems and tissues, and how are you affecting the aging systems? And so it's a back and forth dialogue between what you put into the system, where the system's at in the equilibrium state, and what the system is capable of sort of responding to those stimuli. So remember that as well. Okay. And so in 2024 hallmarks, there is still a clear need to develop new therapeutic strategies now, strategies that are going to improve the quality and the length of life, both, right? The strategies. Now the focus is on strategies. Yes, of course, there will always still be new drug development and new drugs have to be identified, but also, what is the complex interplay between the diversity of the host genome, between chance events, between complex environmental exposures that that individual is being exposed to. Okay. And so then, lo and behold, this extremely complex diagram, but very well thought out, because I want to spend a moment to actually walk through this. And so I'm going to hide this distracting text here. And let's look at this diagram. It's very well thought out. Okay. Let's spend a moment. I really hope people can see my cursor here because I'm starting at the top left of this rectangle. And at the top left of the rectangle, you see cancer cells. Yeah. Then there's the extracellular matrix of those cancer cells, meaning everything that they are seeing, those cancer cells are seeing outside of their cell membranes. What are they seeing? They're seeing stromal fibroblasts that have in turn their own cell membranes and their own signaling happening inside of them. They're seeing this stromal endothelial cells. Endothelial cells are the cells that line the blood vessels. Okay, also vascular endothelium. You have epithelial cells. A lot of solid tumors are starting from epithelial cells. Okay, you have epithelial cells that have not converted and become cancerous completely yet. You have fat cells, adipocytes. You have, of course, the immune repertoire, the immune cells. There's some sort of innervation in or around the tumor. There are some sort of neurons somewhere in that vicinity, at least. Oh. There's the microbiome, whether it's, depending on where the tumor location is, if it's, for example, a colorectal or a GI tumor, then it's very directly close to the microbiome, but also other tumors. The microbiome, as you all know, gut microbiome affects the larger system, the larger human body physiology. And you also have the extracellular matrix, which is not cells, but it's the integrins, the laminins, like the fibers that kind of hold the cells together to make a tissue architecture. The tissue architecture, which then is sort of then organized into organs of the body, right? Okay. Now here, we went through the top, all the cells. Now we're looking at this middle here, where it says microenvironment and macroenvironment. And then they've done this nicely with an XY axis sort of a thing. You have on the Y axis spatial differences within the tumor. And on the X axis, you have time. Over time, there's the primary tumor growth, there's the metastatic spreading. Throughout that whole process, there's systemic interactions that are happening, and there are different treatments that are being seen by the system, right? And so the key point being that it's important to remember the systemic influences that are happening on the smaller tumors as well. This is again just depicting how, there's normal cells, there's early growth and then progression. And then there's escape and metastasis to different locations. But again, food intake, stress, circadian rhythms, sleep patterns, right? Pollution and environment, aging, all of these things are affecting it. And I just want to pause here and say that I would be the last person to try and put the one more thing on people who are already facing cancer, or put the onus on anyone who's faced with something like this, saying, "Oh, you need to take care of this. You need to also take care of this and this and this." Here's the reality, right? There are things that are outside of our control, and there are things that are within our capacity to change. That's all we can remember and act upon as and when we can, and that's also dynamic. But awareness, education about the latest scientific concepts is this. And with that being said, let's look at tumor immunology and oncometabolites. So we went through the 3 landmark papers talking about hallmarks and how tumor immunology would be one component of the hallmarks. And so looking at that, there's so many different types of immunotherapy treatments out there now, right? We have cancer vaccines, oncolytic viruses, of course, immune checkpoint inhibitors that have now been around for quite some time. There's different cell therapy, adoptive cell transfer, sort of single patient type treatments. And there's cytokines. Cytokines are molecules that are secreted by immune cells and other cells, actually, to affect cells in the surrounding through the bloodstream. So, for example, remember I said the outside in and the inside out. One mechanism of inside out is where the DNA in a particular cell produces an RNA transcript that is transcribing something called a cytokine. That cytokine molecule gets manufactured in the cell and released, gets manufactured in the cytoplasm and released through the cell membrane into the bloodstream and goes and affects other cells. Okay, anyways, so when you talk about immunotherapy, there's all these different kinds of treatments that are being studied right now. Some are established through large-scale clinical trials. Some are very early phase. Some are still in the lab, right? But there's all of this. So there's hope, so much hope. Now, just to give you a timeline and an idea of types of immunotherapy treatments over the years, what has been their development timeline. So pre-2010, this is what it was looking like. There was the approval of this IL-2 treatment for malignant melanoma in 1992, way back when. That would still be categorized as an immunotherapy type treatment, but that's all there was in the 90s, right? And then fast forward to all these different approvals. So I just want to highlight here, the material written here is small font, sorry, and it's a lot, but I had to keep it here just to point to you that the boxes with the solid lines indicate approvals, complete like FDA approvals, okay? Other events are shown in the dashed lines. So you have the actual approvals and then you have other sort of milestone events, meaning the phase 3 trials are ongoing, for example. Those are in the dashed lines, okay? The lines with the rounded corners indicate the period of clinical trials. So you can see here in 2017, there was this phase 3 trial of small molecule CXCR4 inhibitor called motixafortide, which is ongoing as of the publication of this paper. And then it also does a good job of specifying the kinds of cancer types that these milestone events are geared towards. So there's all these resources for getting educated about what's out there, but it's a lot. It's a lot.
Kaumudi, I didn't see CAR T, my favorite, on that list.
Oh, yeah, CAR T is should be here, but it's coming up in my slides. Don't worry. I will be
Let me just
mentioning.
ask a time check question
Mhm.
since you
Yeah.
referencing slides. This is unusual for the way we usually operate in that you're lecturing and we're not really having a dialogue.
Mhm.
And I think that's fine because you're sort of giving us a survey of the literature. So I'm letting that roll. But I do
Mhm.
hope that you will come in like a good lecturer by the hour. Are you going to be able to
Absolutely.
come in on time?
Absolutely. And I want to save some time for questions as well. So yeah,
Okay.
that's absolutely right. So these are, the types of immunotherapy treatments that have been developed over the years. And then we are now looking at major immune cell components in the tumor microenvironment. So you have T regulatory cells, okay? You have these mast cells, you have monocytes, granulocytes. All of these are being heavily studied. If you look at this particular environment, the tumor microenvironment can be filled with either cells that are suppressing the immune system or cells that are stimulating the immune system, right? And the tumor itself can can have a relatively more stable genome or a more unstable genome. In the case of a more unstable genome, where there's high genomic instability and mutations, there are more tumor-specific neoantigens that are likely to be released. And then antigen presentation happens, and the immune system can sort of attack the tumor. There are some tumor microenvironments in which the suppressive signaling pathway is on, the Tregs are on, and it's just the immune system, it's hard for the immune system to recognize those tumors. And um, for example, pancreatic cancer is known to be one of those in that category, and then other cancers that are hard for, they make it harder for the immune system to recognize them. And then this is showing you a much more nuanced diagram of the immune component that contributes to tumor microenvironment and how different factors sort of play there's an interplay of the cytokines and the different states at which the T cells or the B cells happen to be at a given point in time around that tumor that'll affect how the immunotherapy is received by the person who is receiving the immunotherapy. So that's an important point to remember about tumor microenvironment. And then lastly, people had asked, so here's a slide about oncometabolites. What are oncometabolites? Oncometabolites are, like the name suggests, they are uh, molecules that are part of metabolism that are typical in an tumor setting, basically, right? So some of these lactate, I mean, they're found in normal cells as well, but their ratio may be changed in tumor cells, for example, in cancer cells. And then you see on the right-hand side the name of the oncometabolite, and then you see the molecular mechanism by which it affects different functions in the cells, like the way the DNA is packaged, which will affect the way it gets expressed into proteins, or the way DNA can be repaired or not, or signaling at the cell membrane, various aspects. Okay, and then what are the treatment possibilities? I'm not going to spend too much time on this. Everyone here has an idea. There's your favorite CAR T cells are also in here, Brad, in cell-based therapies. So all of these different modalities can be used, right, to start that healing process, to continue that healing process. And just remember the evidence pyramid here. Whenever you're evaluating any of these for yourself or for your loved ones, remember this evidence pyramid that you should keep in mind when you're trying to weigh the pros and cons of each of these approaches for the tumors that you're trying to heal through, right? And then nowadays, we're moving towards AI-driven treatment recommendations in precision oncology. So for example, this is just one example of the kinds of work companies and labs are doing to put together. What do you put together? You put together types of biomarkers, the different data sources that they're coming from. So the evidence pyramid that I mentioned, you look at cancer type, you look at the biomarkers, you put all of this information together, and then the hope is that that is going to spit out an evidence-based therapy recommendation or a strategy. and there's going to be, but we're not there yet. This has to be built completely yet. We're not there yet. Just bringing back to mind homeostasis, how everything is in dynamic equilibrium. Remember that even though there's AI trying to guide all these different treatment approaches, there's also all of those healthy cells within the body system of any given person who is faced with the tumorous cells that are also present in his or her system physiology, right? Dynamic equilibrium, remember that. And just one example of homeostasis in health and disease is in gut dysbiosis, or you have a homeostasis in health of the different bacteria that are found in the gut microbiome, and that balance gets tilted one way or the other in various diseased states. So states, away from the healthy equilibrium. Okay, just saying that. And then just a small pause to ponder. I'm not going to say much here. I want time for questions. I have 5 minutes left. Just one deep breath. Some points to ponder. So the hallmarks serve as lighthouses in charting any cancer healing. Education is key. Your community, your healing community will include doctors, nurses, science. It should include science-driven advocates who can help this process, be your partner and help in this. And caregivers, your loved ones, whoever is there, yourself as a self-advocate. Remember that your human body is a community of cells. It's a community of cells that embodies your own meaning. And with the right tools, that larger community of cells can redirect and destroy the dysregulated community, a smaller subset, which is cancer. Even in the presence of the most advanced, widely metastasized cancers, there's still significant portions of human tissue that are trying to behave as if they would in absence of cancer. And also, please try your best to keep track of any tissue that is collected post-diagnosis, and please try to stay informed about how that tissue gets used. There are times when you might have to educate everyone involved about how you may wish to access that tissue for future use, for biomarker testing, for any of that. Remember that. It's necessary to have a weighted plan, if then sort of a plan for healing through this process, integrating as many components as you can of the healing process. Generative tools can be helpful here, AI, but also when used alongside consultations with educated professionals, such as doctors, nurses, and science-driven advocates. And with that, if you ever want to together find YOM, here's a short survey, small form you have to fill in on our website. It's called yomwellness.org. You can go there, and that's it. I'm ending there. So I can stop sharing.
Thank you, Kaumudi. Again, because we only have a couple of minutes, what I would encourage us to do is take your slides and publish those as quickly as possible. Maybe we could do them on our Circle forum, and then people could engage with you in the kind of question and answer we would normally have here on that platform, so that everybody can see that conversation. And then we will also release the video very soon, in a matter of days. Getting the transcript and the notes together will take longer. But for those who want to dive into this, we can solve that way. I almost hesitate to try to shoehorn questions into the remaining 2 minutes, but Roger, your hand's up, so you get
Well,
the chance.
Since we've only got a couple minutes, the big question, Kaumudi, is this was a master class right here, and obviously it's so much more information than you can fit into an hour. What is it exactly you're doing now? Is this an educational course? Is this personalized one-on-one counseling? I'm kind of curious as to how you're going forward going to fit into the community.
Thank you, Roger, for asking that question. So this, yes, absolutely, you're right. This was like a master course with so much information. But this is to say that if anyone would like to go through this sort of information in a slower manner pertaining to their own personal situation with their cancer specifically, then reach out to me, fill out that form, or just, Yom Wellness, yomwellness.org. and email me. I put my email address here. So in the chat, and we can have that conversation, and hopefully I can help. So yeah.
Okay, thank you.
Kaumudi, how do you bridge? So I'm just going to be very simple-minded here. What you presented in the content, we had Michael Castro on, and he had molecular pathways, and they were very, very complicated, complex, as you shared as well. So that's super microbiology, detail, scientific, evolving, hard to understand, complex, detailed. You almost need a PhD to be able to actually follow what you described there. On the other hand, now you're talking about yom wellness, and it says like take a breath and reflect for a moment, and your cells, some of your cells are still healthy. I find a disconnect between those two things, one of which is very deeply science and scientific, and the other is almost at a level of wellness and emotional health. How do you reconcile those two things, which seem to me to be almost incompatible?
Thank you so much for asking this question, Brad, because healing has to be a part of medicine, but the way the system is set up, it's not always possible to have that, to integrate the very sort of reductionist approach that science has taken all these years, that has gotten us very far in terms of technological innovation to have these latest and greatest treatments. But now that the time has come to integrate all these sources of information, even a scientist who's really hard at work trying to discover something new has to ask the greatest scientists out there, not me, ask the greatest scientists out there. Even the best scientist has to take a pause, a moment, a deep breath. And it's only in that space that that information can get synthesized. And true genius or true creativity or a true path forward always stems from a moment of core silence. Once you've done all this research, all of this information, to synthesize that, there has to be a process. There have to be tools for centering. And sadly, in today's fast-paced world, with the way the system is set up, there's not any designated time given for that. It becomes one more thing that the person dealing with cancer in their family has to create for themselves, is that space and time to go within, synthesize, take that one deep breath, because it's a lot of information. But for synthesis, bringing it together, we need this sort of mindfulness or going within sort of a tool. They're tools. And so that's how I reconcile the two parts, if you will, is there's that, yeah. As you go outwards, you gather all that information, you verify it, you look at the evidence pyramid, you kind of weigh the pros and cons, take that deep breath, get into your core, remember, you have this community of cells, which has a meaning, which is you, which is so much larger than that smaller subset community of cells that's trying to derive its own meaning in its own manner with its own molecules and distorted, dysregulated sort of manner. It's using the mechanisms that are known to be present in embryogenesis. That's what tumors are doing. So that's how I reconcile the two parts.